thank you to the organisers as well for the opportunity to talk today. I'm tremendously excited about talking about my or our research, but also quite apprehensive. Doubly so now I've been given 2 microphones, so I feel like I'm in some kind of FBI interrogation or something. But no, I'm apprehensive because when we developed our modelling approach, we completely disregarded anything to do with dynamic energy budgets, being naive to it in the world. So you'll not really see it in the talk at all. I've already apologised to Bas for taking such a grievous step. But what I hope to do is to convey what I think are the values of such models, or the value of such models for potentially for linking dynamic energy budgets to broader aspects of ecological modelling, and also for putting ecological modelling on an equal footing with other more physically based modelling, for example, climate modelling. So I start off by just summarising one of the most striking things about the planet on which we live is the diversity of life on that planet. And the diversity of that life and the ecology which it undergoes is tremendously complex, but it's also fundamental to making the planet habitable for humans and for other life forms on the planet. And in the present day, habitat conversion, overexploitation, climate change, pollution, and invasive species are acting simultaneously to affect biodiversity in unprecedented ways. And so, at least it's my personal view that we're in a situation where it's critical for us to get new tools so that we can understand the impacts of our actions now and into the future, both to understand the problem, communicate it to other people, and effectively guide the resources, the limited resources which we have to tackle the problem. But perhaps in part due to the complexity of ecological systems, I know I'm sort of probably preaching to the converted here about mechanistic modelling as opposed to statistical modelling, but my view is that particularly much of conservation modelling and also a large part of ecological science relies on pattern-based models or statistical models or phenomenological models, however you want to describe them. So, for example, there was a colleague of mine at WCMC, Tim Newbold, had a paper in Nature a couple of years ago now, which was a tremendous piece of work, but essentially is a fairly simple statistical model applied to a huge data set of biodiversity responses to different land use changes. So here, this is is that me? This is the relative species richness. Am I pressing the wrong thing here? It's just the red button.
No, just,
okay.
Yeah.
Yeah, okay. So this is the species richness change relative to the primary vegetation state for a set of human-modified habitat types. And the general story is that human modification causes a decline in the species richness, but also in other properties of biodiversity in different locations. But of course, there are problems associated with correlative models. So they often can't diagnose causality in those relationships. So we see here for this mature secondary vegetation class, actually the species richness has increased as a result of human modification. And that data set alone doesn't tell you about what's caused that increase. Perhaps the bigger concern is the extrapolation or applying these models to project changes in new geographic, temporal, or environmental spaces, which is particularly an issue in the present climate of global change, where we have these simultaneous forcings affecting biodiversity in novel ways. But I make the argument here, I draw the analogy with climate modelling, where the climate system is also complex, right? There's a lot of internal feedbacks and a lot of complicated processes. I'd say it's an order of magnitude less complicated than ecological systems, but still it is a complex system. But these guys have been developing mechanistic models for over 60 years. So starting way back in sort of 1956, where Norman Phillips published his paper on the general circulation of the atmosphere. Since then, climate models have been developing, and then for the last at least 20 years, we've had the era of the Intergovernmental Panel on Climate Change, which can draw upon a suite of mechanistic models to allow them to understand the future of the climate system and also understand the implications of different human mitigation options. And of course, alongside the development of climate models, there's also been the development of data required to make them better. So in the present day, we have a suite of essential climate variables, a lot of them remotely sensed, which have been, in a large part, they've been instigated or put into place as a result of the demands of climate models to be able to make better predictions about the future of the climate system. I think that we are in this concept of general ecosystem models, that we're in a similar position to where the climate modelers were back in the 1950s. So there was a lot of skepticism about whether or not there ever would be a general climate model. So Bert Berlin, who went on to become the chairman of the Intergovernmental Panel on Climate Change, basically said there's very little hope for us ever building such models. But then a couple of years later, Norman Phillips produced his first model on this minuscule computer with 5 kilobytes of memory. But the key thing here is that it was a simple model. It was a simplified model of the world. And we went on then to produce more complicated and sophisticated models. And the Earth system science community is where it is now as a result of these kind of efforts. So I'd ask you to just bear in mind that I think that we are, or at least our research, is very much in this simplified world. So it's a nascent effort. The FBI are listening to me. So to summarize the general philosophy of the modeling that we've been trying to do. So we want a model that is globally applicable and is spatially explicit, that can couple the terrestrial and the marine realms. Importantly, it's trying to give a balanced consideration to all trophic levels. So we're trying to represent all life on Earth, but give it a fair representation in the model. We want the model itself, the code, to be transparent, open, and reproducible. And that's always probably a subjective measurement, but we've tried as far as we can to meet these criteria. But we want the model, we want to specify the model at the level of individuals because they are the key agents in which ecology happens. And we want to constrain the model to processes which apply to individuals and allow emergent properties to emerge, for higher levels of organization to emerge. And the key point I'd make here is that I'm not proposing this is the only way you do this modeling at all. I'm just suggesting this is one way, and this is the way that we've been pioneering to do this. And so we published a kind of proof of concept of this model back in 2014. So the model works on a grid-based approach. So we apply a grid that can be global, it can be flexible in extent and its resolution. Within that grid, we represent a realistic geography, so the distribution of land and sea is realistic. We apply realistic environmental conditions, so air and ocean temperatures and precipitation, for example, taken from climatologies for the contemporary world. Most of our effort has been in modeling the animal part of the community, because obviously there have been a lot of efforts on modeling plant dynamics historically. But in each grid cell in the model, we do implement on the terrestrial world a mechanistic data-constrained carbon cycle model, and that provides the vegetation resources on which herbivores can feed upon. And in the oceans, we use remotely sensed measurements of phytoplankton to provide an estimate of the planktonic biomass available for herbivores to feed upon. But most of what I'm going to talk about today is on the animal part of the community. And what we assume is it's an agent-based model, and what we're assuming is that we place agents within a grid cell, and for the duration of a time step, those agents are constrained within that grid cell. We assume that they're well mixed, so they cover the whole of that grid cell, and they can interact with anything else in that grid cell. And then between time steps, they can potentially move between them. So I mentioned before that we want to build the model at the level of individuals, because we want to model all individuals on Earth. But we face two key problems with that. First of all, if we wanted to represent organisms as a set of species, we haven't even described all the species on Earth. So it's quite problematic to do that. So what we do is we resolve organisms according to functional groups. We define functional groups according to a set of key functional processes. So every organism in the model has a set of hard functional traits. So they have a feeding mode, they have a metabolic pathway, a dispersal mode, and a reproductive syndrome, or if that's the right term. But there'll also be, and these bold traits here, they are fixed for a functional group. So organisms belong to a functional group determined by these, but organisms also have traits which are fixed throughout their lifetime. So they have a juvenile mass and they have an adult mass. So the juvenile mass at which they're born, the adult mass at which they become reproductively mature. But then they'll also have an ontogenetically varying mass. which is obviously varying through time. The second problem we face is that just computationally it's unfeasible for us to model every single individual as a separate agent in the model. And so we make this abstraction to say instead of modeling individuals separately, we're going to group organisms together into what we call a cohort, which is a collection of individuals with exactly the same traits, but then also with an abundance of those individuals. So I'm going to summarize now how those animal agents undergo their ecology in the model. So if you were to take this blue circle representing a cohort, the black dots within it represent the number of individuals in the cohort, and then their diameter might represent their body size within a time step. So within a time step, all agents within a grid cell, or within a model rather, undergo all of these ecological processes. And I'm hoping that this comes out as being something analogous to the energy budgets paradigm. So organisms metabolize, which means to a loss of their biomass. They're burning off biomass to provide energy for their maintenance. And we model that as using the metabolic theory of ecology. So primarily as a function of their body mass, but also of the ambient temperature for ectotherms. For endotherms, we assume that they thermoregulate at 310 Kelvin. We also introduce in this component here a critical temperatures for ectotherms. So when the ambient conditions are outside some optimal, some temperature range, then ectotherms' ability to be active is reduced. So there's a temperature dependence on the metabolic rate. Organisms might all be predated upon by another cohort, which would lead to a loss in the abundance of this particular agent, and the abundance of this agent would be turned into biomass of the prey individuals, and there's some possibility that this cohort would eat itself, either from other animal cohort or from some plant matter, and you'll see from this that we model predator-prey interactions using a Hollings type 3 function. So we have this logistic curve, where the amount of prey abundance which you eat is a function of the prey density. But there's also each predator cohort has a preference for a particular prey size, which is focusing on that, affects the attack rate, which moves this curve backwards and forwards along that curve. I'm just not sure I'm doing anything wrong, but I apologize if so. So if a cohort is successful in assimilating enough biomass to meet its metabolic costs and grow through time, if it reaches its maturity mass, it might... Hello?
I'm just going to switch off the volume on the computer.
Okay.
So you will not have the noise.
Cool, thank you. You haven't switched off the volume on me, have you? No. So if they reach their maturity mass and they can start allocating food that they've assimilated to a reproductive pool, and then they might be able to spawn an offspring cohort into the model, which would then be a separate agent going about its own set of ecological processes. There are a set of non-predation mortality terms applied to this cohort, so old age, senescence mortality rather, background mortality and starvation mortality would apply to it. And then there's also some probability that in the current time step, the cohort might move from its current location to another location. Again, one of the key parameters for all of this is the body size of the organism, but then the functional traits of the hard functional traits of the cohort determine qualitatively how it does some of these processes. And I think the scheme that we've come up with fairly independently is very analogous to this schema, which is Sibley et al. Published back in 2013. So food comes in, so the predator-prey interaction occurs, for example. Food is assimilated, so the combination of these two. It gets allocated primarily to maintenance and meeting metabolic costs. Any subsequent biomass assimilated goes into growth. Subsequent to that, we might allocate to reproduction. And what we don't have explicitly any energy reserve in the model, but we assume that organisms can lose some of their body mass if they're starving, but that the starvation mortality rate increases exponentially as you move away from your maximum achieved body mass. So if you shrink too much, basically you're dying out of the model. And so What we do is we throw into the model a whole load of agents with different sets of functional traits in a relatively neutral manner into each grid cell in the model. And what I'm showing here is a visualization of what's going on within a cell over time. So those cohorts that we throw in interact with each other according to the rules which I've just described. And what we've got here is a single grid cell, the output from a single grid cell, where each point on this plot represents a different cohort. They're color-coded. I hope you can see the colors. The green is for herbivorous cohorts, orange is for omnivorous cohorts, and red for carnivorous ones. They're organized on a y-axis, which represents their current body mass, and an x-axis, which represents their adult body mass. So you see the cohorts, they never change in their x-axis position because that trait is fixed throughout their lifetime. But the cohorts will be growing upwards. They'll be increasing their current body mass, typically, going up in that direction. And here, the axes are log-scaled, and here are just some, although cohorts are not species-specific, right? They're sort of heterospecific in their identity. These are just some examples of the organisms which tend to have this kind of body size. So organisms down here tend to be the size of centipedes, although these are herbivorous ones. Whereas up here, we've got elephant-like organisms in the model. And when I play the simulation, hopefully it works, you'll see 12 months worth of data happening. Can I get it there? Here we go. So no. Terribly executed. So you see cohorts are, generally they're growing upwards, they're increasing in their body size up this way as they interact with each other and grow. What we can look at also in this visualization is that, so the trophic interactions in which are happening in the model are evolving dynamically. So if we were to click on one of these cohorts, in this case we're going to choose an omnivore cohort, we can see that these are the cohorts on which it's predating upon, and these are cohorts which are predating upon it, and these are changing through time. And so the key feature you'll see is that on average, so the size of these circles represents the abundance of each cohort. You'll see that typically the smaller body-sized cohorts have a greater abundance compared to the larger body-sized cohorts. So that's what's happening within a single grid cell. But then we also essentially have that same process happening in every grid cell in the world, and then those grid cells communicating with each other through dispersal. And this is not particularly informative, but it does show you the dynamics of the model. So we start off with this neutral distribution of biomass. Everything gets the same amount of stuff everywhere, and it's distributed in a fairly random manner. That's not what I wanted to happen. And fairly quickly, this is showing the color code here is the total biomass of animals in any grid cell, and each time step is a month of time elapsing in the model. So red is where there's more biomass, and blue is when there's lower biomass. And so what we can see is that fairly quickly the model settles down to capturing seasonal dynamics in the total biomass of animals in different environments. We tend to have more biomass in tropical regions and more biomass in the upwelling regions of the oceans, which are varying dynamically through time. But what we'd like to do is compare the model against some real-world data and find out if we apply a pattern-oriented approach, does the model look like what we see in the real world? And when we do that, I think it's, if you can have your own opinions, but I think given how crude the model is and compared to the real world, it looks, to my mind, broadly realistic. So here we're looking at the level of individual organisms at the growth rate of those individuals as a function of their body mass, and at their lifetime reproductive success as a function of body mass, where the grey points are model predictions from terrestrial and marine cells, and the black points are empirical data derived from the literature. What we find is that for growth, there's a very, very close agreement between the log linear growth rate, or increasing growth rate as a function of body size between the model and empirical data. There's the slopes of these two, the model and the empirical data, are not significantly different in this plot, but there's obviously far greater variability in the model predictions of lifetime reproductive success compared to empirical data. And partly this represents a difference in what we're predicting here. So this is lifetime reproductive success of approximately individual organisms, whereas these are species-level averages of lifetime reproductive success for a given species in the real world. And so my conjecture is that some of these estimates are averaging out variability in the within species case. And now if we look at collections of individuals in a community, we're looking at here the abundance biomass distribution, so the numbers of individuals distributed as a function of body mass for carnivores, omnivores, and herbivores. And if we were to take the slopes of these relationships, they are broadly log linear, which agrees with some empirical data and not with others. But if we look at the slopes of these relationships, then they fall within the bounds of some of the estimates of the slopes of those relationships derived from empirical data. And then this is kind of a mega plot, but what we're showing here is trying to compare ecosystem structure, the structure of whole ecosystems predicted by the model compared to empirical data. So these are a set of sites where there's been comprehensive sampling undertaken to try and understand what's the total biomass of different trophic levels in that location. And we're comparing here the ratio of the animal biomass to the plant biomass for different trophic levels, green being herbivore, blue omnivore, and red carnivore, where the open symbols are the model predictions, with the symbol being the median of an ensemble of 10 simulations, and then the line being the maximum and minimum values. And then the closed symbols are the empirical data with some uncertainty, again, the maximum and minimum range, where there was more than one observation available. And you can see these top row are all marine systems, or I should say they're coastal systems primarily, and the bottom row are all terrestrial systems. And there's an awful lot of stories in here, but my take-home message is that the model does quite well in some places and really badly in other places. So for example, if we take the Etherne estuary in Scotland, we see pretty good agreement between the model and the empirical data. The Ems estuary, there's a good agreement for herbivores and omnivores, but not for carnivores, whereas for other locations, for example, Santa Monica Bay and the Schwarzkopf estuary, there's quite a big difference between the two data sets. In the terrestrial realm, we can pick out some characteristically the Serengeti and the Cedar Creek ecosystems where the model and empirical data agree pretty well, but in other locations like the Arizona and this forest system, there's a huge amount of disagreement between the model and empirical data. And so clearly clearly the model is very crude compared to the real world, so I'm not surprised that it's not doing a very good job in some locations. What is probably more surprising is that it does a decent job in any of the locations at all. So what we think is that for these more grassland-like systems, that's more in keeping with the sort of assumptions of the model, the fact that all plant matter that's non-structural, non-woody is available for animals to eat, for herbivores to eat, and also that organisms are well mixed within that environment. So both of those assumptions are not true in forest ecosystems and also in more desert-like ecosystems where plants are defending themselves. The other point to mention here is of course, that all of these data, although they're taken from what we think are possibly more pristine sites, they're also obviously subjected to anthropogenic change. So we're running a model of a sort of pristine world, but comparing it to human-impacted data. So I wanted at this point to move on to how we've been trying to use the model to understand sort of some ecological theory, and particularly this idea of mechanistic macroecology. And I'm going to use the example of Bergmann's rule, which I'm sure you're all familiar with, the latitudinal gradient in body size, particularly for endothermic organisms, as illustrated by this, the data from this paper published last year, which is showing the frequency distribution of mammals with different body size classes, where the pink points here are the body sizes of species which have gone extinct or had their ranges modified by human effects. And you'll see that in high latitudes there are far more larger-bodied organisms than there are at low latitudes, and so the mean body size of these communities are likely to be larger than in low latitudes. Excuse me. And there have been a whole set of mechanisms proposed for why this latitudinal distribution emerges. But what we're excited about is that if we look at the model predictions of community mean body size for endotherms and for ectotherms, and we plot a latitudinal profile, so taking the statistics across each longitude, across longitudes in each latitude band, we end up with a size distribution across latitudes that is consistent with an empirically derived pattern based on species traits and range maps. And we see that for endotherms, we see higher body-sized organisms in the poles and also in arid environments, particularly the subtropical desert regions. And we see some evidence for both of those patterns in mammals, less so in, for birds and less so in mammals. We also see evidence for an inverse Bergmann relationship in ectotherms, and there's been some equivocal evidence for that in empirical data, but I don't think that's as clearly as well established. But what's exciting then, if we produce that in a model, is that we can perform some environmental manipulations to try and test those mechanisms for why those body size distributions emerge. So what we can do is we can alter, for example, latitudinal gradients in the annual mean productivity, or in the seasonality of that productivity, or indeed in the temperature or the seasonality of temperature. And this is just showing the latitudinal profiles of those conditions in the full model, as well as the sort of annual mean conditions or the zero seasonality conditions. And then this is showing if we set every cell to have the same annual mean, the same value, the global mean annual productivity, this is the deviation in productivity that results. And what we find is that here I'm showing you, for a simplified model, the latitudinal variation in the median across latitudes in the body mass of endotherms, and the proportional or the relative deviation in body size as a result of performing these knockout experiments. And what we find is that in the northern hemisphere, the total amount of productivity is the most important for decreasing, or at least the greatest decreases in body size of endotherms, particularly in high latitudes. Whereas in the southern hemisphere, it's really the seasonality of that productivity that seems to be important. But there is some effect of the seasonality of temperature. If we remove any seasonality of temperature, we end up with smaller body-sized endotherms and all. And what we find, if we look at the behavior of individuals in the model, we find that in an environment with global annual mean productivity, so more productivity in these environments, smaller organisms are able to assimilate more, reproduce more, and have a lower starvation mortality term applied to them. Sorry. If we look at the ectotherms, then we see sort of equivocal evidence of the effects of these three knockouts, so the effect of taking out total productivity, seasonality of productivity, or the annual mean temperature. But we see a very strong effect of this removing the seasonality of temperature. So that leads to a big increase in the size of ectotherms, particularly in the high latitudes. And the really cool thing for me in this, I realize that this is obviously a simplified version of reality, but what this is, I think I alluded to earlier, endotherms are assumed in the model to thermoregulate themselves no matter what the ambient conditions are. So they're not responding directly to changes in the temperature in the model. So this is a trophic interaction effect, or at least a resource interaction effect. Ectotherms are responding to the changes in seasonality of temperature, and that's having a knock-on effect on the endotherms. So there's some kind of bottom-up limitation on the endotherm body size happening as a result of the trophic interactions in the model, which I think is, at least to my mind, is pretty interesting. So, how many for time? Good. And I want to move on to talk about how we're thinking about using the model, or these types of models, to understand responses to global change. I introduced this slide earlier, which is this predicts model of the responses of biodiversity to different human-modified land use, or the transitions in land use as a result of human modification. So what we wanted to do was in order to evaluate whether the model is doing something reasonable in modeling responses to global change, for example, land use change, we need to compare that to data. But there aren't many locations in the world where people have quantified the total ecosystem response to land use change. And so what we ended up doing was taking this statistical model so very closely associated with data, model for how ecosystems might respond to land use change, and then comparing the predictions from that model to, which is obviously completely free of any mechanism, to our mechanistic model of the response to land use change. And we conducted this for the whole of South America. And what we find is actually that the median responses, so this is Madingley and these are the predict responses, this is for the total abundance of all organisms in ecosystems, that the both models predict very, very similar median responses to land use change across South America. So it's about 10% for all organisms, about 8% for endotherms, and nearly 10% for ectotherms. But clearly you can see there's much greater variation in the response of ectotherms, particularly in Madingley, than in predict. And we're going to explore that in more detail. But the point I wanted to make is that we have some belief that the model, at least if we compare it to another model, the model might be doing something plausible in how it responds to global change. So now what we're really interested to do is, particularly within the framework, for example, of planetary boundaries, where we're trying to understand how far can we perturb different ecosystems. Do different ecosystems, or in different parts of the world, do they respond in the same or in qualitatively different ways to the same impact? So here we're modeling the conversion of pristine ecosystems to human-modified ones. And we do that in the Madingley model by human appropriation of net primary productivity. So in other words, we're assuming that some of the productivity that would have occurred in the natural system is being taken out and used by humans instead of the natural ecosystem. And the experimental setup is to run the model for 1,000 years until it reaches some quasi-steady state for a whole set of different ecosystems, and then to increase the amount of productivity that's being taken out of the system by 1% per year, and then until it reaches some threshold, which is a set of deciles from 10 up to 100%, and then hold that constant for the duration of 100 years. And then we want to compare the system after that 100 years has elapsed to this pristine state and see what the effect of that perturbation was in different ecosystems. And what we find, which is really cool, is that different ecosystems, here we've got a high productivity aseasonal environment, and a high productivity seasonal environment, and then two lower productivity environments, a seasonal one and then an aseasonal low productivity one. They have qualitatively different responses to the different land use changes. So in this high productivity aseasonal environment, what we're showing here, is the relative biomass compared to the pristine case of these different perturbation levels rather. And the points are coloured with grey being autotrophs, green being herbivores, and blue omnivores and red carnivores. You can see that the plant biomass is declining almost linearly. So as we're harvesting more biomass from the system, the plants are declining linearly. But the biomass of animals is actually quite insensitive until we reach levels of about 80% appropriation. And then suddenly we see a sort of collapse of the ecosystem. The total biomass is declining quite rapidly. Whereas in France, the whole system is declining linearly as we harvest biomass from the system. And indeed, in the Gobi Desert, the animals, the herbivores and carnivores, are responding sublinearly. So there's a greater decline in those than there is in the plant matter, and so on. So the key point is that even in a crude model of the ecology of the real world, we're seeing qualitatively different responses in different ecosystems. The next question we'd like to ask is do these systems return to their pre-impacted state if we allow them to recover? So again, we've got the same experimental setup. We run the model to a quasi-steady state, impact it up to a high degree of perturbation in a linear fashion, and then decrease it in exactly the same fashion and allow it to recover, and then compare the model here to how it was here. And what we're showing here is for those very same locations, this is the trajectory of biomass, where the solid line is this outward component. So the solid line is when we're going up in perturbation, and the dashed line is where we're going down in perturbation. And so for the Ugandan cell, we see actually we return to approximately the same levels as the initial conditions, whereas for France, we see a small deviation. So we go out in terms of carnivores, we start at over 10,000 kilograms per kilometre squared, we impact it, and then when we return with a lower biomass than when we started to. And in these other cases, we end up with greater differences. So there's qualitatively different capacity for these ecosystems to return to their pre-perturbation state as well. So I think I'm on time, which is amazing. So I wanted to finish up by talking, to my mind, what I think are some of the key challenges facing the development of such general ecosystem models. And in no particular order. So a key one is data. So one of the critical issues is that almost all the data we collect in the present day obviously has human impacts associated with it. And so in order for us to compare the model to that data, we really need to be modeling human impacts as well as the pristine ecology. But essentially more data on ecosystems, broadly speaking, and more representative of all taxa, but also spanning whole ecosystems in different locations would be critical for doing this. Of course, there's our understanding of ecology and how we improve that or incorporate that into the model, or which bits of ecology do we need to improve upon to make better predictions, and we don't know that objectively at present. I think a critical concept that we don't really address enough in ecology is how predictable are ecosystems anyway. This is just a question which I pose my undergraduates when I'm lecturing them, which is, if I was to ask them to predict the future state of a jar of sweets in the lecture theatre, would they be able to do it? And they can come up with some simple models for how they might do it. But then if I was to tell them that the sweets were interacting with each other and they were trying to murder red sweets, or that they were reproducing new sweets, or indeed even if they were evolving, would they be able to make predictions about that jar of sweets? And then clearly the answer is no. Ecological systems are intrinsically hard to predict. But some features of them might be predictable, and that's the bit that we need to be focusing on. And then lastly, clearly there's a question of both computational and human capacity to use computational devices. But I wanted to finish on a couple of slides, which I think, these are issues which I think are critical for, at least for our model development, possibly also for dynamic energy budgets. So at the minute, the models that we use are devoid of any intelligence of organisms. Organisms are just following these deterministic rules with some stochasticity applied to them. They're not, oh, by the way, this is some representation of animal intelligence. I don't mean that exactly. I'm using a broad term for intelligence. So animals obviously perceive a near-term local environment. They have embedded in them some evolutionary hard coding to do things the way they do. But how do you represent that in models? And that is I think, a question which isn't really broached enough in ecological modeling, and could be critical in the face of global change as their sort of evolutionary learned conditions are changing. The example I gave is, so for example, if you're an organism in high latitudes and your resources start to deplete through time, you've got a set of decisions facing you. You can choose to move somewhere where there are more resources, or you can start to downregulate your metabolism to try and hibernate or go into torpor for the winter. And how do you make that decision? What is it that's operating there? And then this for me is our kind of next frontier, which we're just working on at the minute, is to take a whole model and put it within either a formal Bayesian framework or indeed within an approximate Bayesian framework to bring the model closer to data so that we can test objectively the model structure, to which bits of ecology are the most uncertain and which particular parameters do we need, provide the most of the uncertainty in the model, which therefore might target what data sets we need to improve or even which ecological functions we need to incorporate into the model. But also might allow us at some point to be able to make predictions with some uncertainty associated with them. And with that, I'll say thank you very much for the opportunity to talk and for listening.